Hello, everyone. And first and foremost, I'd like to thank the organizers for inviting me to participate in this online virtual seminar series talking about immune oncology. Today, I'll be talking about B cells inside and outside of tertiary lymphoid structures and how we can target them for improved immunotherapeutics. My name is Tullia Bruno. I'm an assistant professor of immunology at the University of Pittsburgh and at the UPMC Hillman Cancer Center. If you want to get in touch with me after today, my email is listed on this first slide. So one thing that I like to set the stage with is looking at a picture of a secondary lymphoid organ, such that's found in a tonsil or a lymph node in our bodies, and what helps with general infection and response to infection. And so these yellow cells that you see that are in the very circular structure, this is called a germinal center. This germinal center is the site of T and B cell education. It's where high affinity antibodies are made and where a lot of proliferation and crosstalk between these cells occur. And so they're very important for optimal education and optimal responses to things like flu and bacteria when we get infected. And so we take a lot of cues about what's happening in these secondary lymphoid organs, not only for B cell biology, but also for tertiary lymphoid structures that form in tumors, in these solid tumors at the site. And that's going to be the main focus of today and thinking about what's going on and how we can target these tertiary lymphoid structures. But also, I'll have a little bit of data at the end talking about what happens with B cells when they're not within that structure, because I think that's another important angle to consider. Okay, so I would like to highlight this as a first and foremost is that the cancer immunity cycle has been updated. Really, we think beyond T cells now, and a big thanks to Dan Chen and Ira Melman for this update of the cancer immunity cycle that has come out over the last couple of years. And so what you can see here is that tertiary lymphoid structures have been added into this cycle here and really thinking about interaction of immune cells. And so that's something to keep in mind is that I think when we think of a germinal center and a secondary lymphoid organ, particularly for a B cell, we think of high affinity antibodies. But within a tumor, tertiary lymphoid structures often don't have germinal centers, which we'll talk about today. And so that can't be the only function. And so I think these interactions, this antigen presentation, this survival within these hubs is quite important. And so a question to keep in the back of your mind, which we often get asked, is if you have immunity in your lymph nodes, why do you think you need local B cells and tertiary lymphoid structures to help with that immune response? And so it's just something to think about as we go through the presentation today. So one piece of background to think about is that TLS have been cited in multiple diseases. And in fact, in autoimmunity, it's been shown that these structures are quite important in exacerbating a disease. So they form these germinal centers, and patients often then have worse disease as a response of having these structures. But in general, they help with homeostatic balance, right? So in a flu infection in your secondary lymphoid organs, you're going to have the infection, and then that lymphoid organ is then going to not be as active once the infection is cleared. And so it keeps a homeostatic balance in your body as you're fighting an infection, but also as that infection is coming down. So in cancer, we're still learning a lot about these structures. We know that they don't look exactly like a secondary lymphoid structure, and I'll show a little bit of that today. But we also know that they've been more prognostically beneficial for patients than they have been in disease settings like autoimmunity or transplant rejection. And here are the data that really, I think, brought TLS back to the forefront of immune oncology. So there was a series of papers in 2020 that is highlighted here in a news and views that I was invited to write for those papers that really demonstrated that having these structures in patient biopsies or tumors at baseline really then correlated with better response to immune checkpoint therapy. And this was primarily PD-1, CTLA-4, or a combination of the two. But what was striking about this for me is that it wasn't just in the context of metastatic melanoma where we would suspect, yes, these patients have other biomarkers of response, but it was in more canonically quote unquote cold tumors like renal cell carcinoma or sarcoma. And so this was a really important time, I think. And I think pathologists have been seeing these structures for years, but really now we're seeing some sort of a link to an immunotherapeutic response. So that definitely piques the interest of even people that don't work on these. And the other pieces of evidence that we have from humans is that when you have these structures present, you usually get more CD8 T cells that flux into the tumors. And those CD8 T cells are often more functional. They have less of an immunosuppressive access. present, such as the CD8 Treg axis. And of course, B cells and T cells are more functional, as I've already mentioned, when they're within this structure. And I think a lot of these studies that we've seen in humans, because that's where a lot of this was grounded, has now been translated to physiologically relevant mouse models that are coming online. And I won't have time to talk about that today, for the sake of time, but happy to talk about it offline because my lab and other groups have really started to generate models or use or find models that are best for studying TLS development and modulation. And so on the right-hand side of the slide, what you can see is a tertiary lymphoid structure. This is done by multispectral imaging now. For the most part, you can, triage samples for the presence of a structure by just doing a singleplex CD20 and looking for the structures by B cells because B cells are a key hallmark of the structure. But most people then move on to multispectral imaging. And really having B cells and CD4 T cells aggregate together is step one. But then step two is then having these high endothelial venules that allow for these cells to traffic in. And then a lot of times you have to have a follicular dendritic cell network for a germinal center to form. And we're going to talk about the different states of TLS in a minute. And once that germinal center forms, you can have activity of that germinal center via Ki67 and AID, which looks at proliferating lymphocytes or B cells going through somatic hypermutation. And what I'll mention here is while we measure this out in the germinal center, you can also measure out these activity parameters in the full structure. And I think it's important to do so because it's not just about the state of the structure, but it's how functional that structure is as well in the microenvironment. So the question then becomes, with this background, how do we maximize B cell and TLS function within tumors? And so the reason that we want to do this, I think, and just to give a little bit more background before I get into some data in the lab, is that we have found that we might be able to select patients for therapeutics via their TLS. And so this paper here that I'm highlighting is from my French colleagues, that after we saw that patients were more responsive when they had structures in their baseline tumors to these immunotherapeutics, they then went on to sort patients based off of their TLS signature. And then by sorting those patients for clinical trial, they saw that patients did better once those patients that had TLS were bucketed into that trial. And so I think this is very telling that it can be used for patient selection. But I think as we do this, we have to learn more about what's happening with TLS dynamics, both in humans and in mouse models. So I hope that I've convinced you that these are important in these background. I think they really are multifunctional. I want to hammer that home. It's not just about antibody production, right? It's about all these other functions that you see on the left-hand side of the slide. And I think there's a lot of ways in which we're targeting different cell types and different axes to improve these structures. And there was a really beautiful review in Nature from some of my colleagues that you should check out on the right-hand side that talks about different ways in which we are trying to modulate structures in patient samples, but mostly also thinking about modulation in mouse models and then translating over to patients. Okay, so this is all well and good, but where are the problems or the issues? So it's not necessarily just the tools that were lacking for a very long time, which we're now getting those online, but it's more about the number of cues and the number of cell types that are really involved with TLS to form properly. So TLS have to have chronic inflammation and chemokine production. They need to have quality antigen, and that antigen has to persist. And so having, antigens like HPV or having microbiota, those antigens have been shown in the literature by myself and others to drive TLS formation, particularly getting to a germinal center. But I think just having the right antigens in general will help for these structures to thrive. And one other thing that I'll talk about today is the stromal cells are very important. You have to engage the stromal cells and they have to be activated and helpful and not suppressive, particularly because these structures, while they can form at different sites of the tumor, on the tumor edge, in the tumor, they always gravitate towards the stromal pocket. So that stroma has to be there. The other complication is that TLS can be modulated by multiple things: tumor microenvironment, the initial site of the tumor microenvironment, like the lung versus the breast, whether it's a metastatic site, but also therapeutics. And I'll show a little snapshot of this today to kind of show you where we're going and what we're thinking about with this. The other complication is the state of the TLS. So while we've been talking about TLS in general, there are 3 accepted states at present early lymphoid aggregate, a TLS without a germinal center, and a TLS with a germinal center. And naturally, as you have more of a germinal center, you have more activity of these structures. And so we'll talk more about this in the context of our ovarian cancer studies, but this is also an important point because any one patient doesn't have the same type of TLS. They have different TLS types. Sometimes they're more predominant for a germinal center, not often. Sometimes they're more predominant for a lymphoid aggregate or a TLS without a germinal center, which is mostly the norm in treatment-naive patients. And so to summarize here, TLS state is complicated. TLS factors are multifactorial, and the modulation plays a key role here. So today I'm going to talk mostly about treatment-naive studies in the lab, as we're getting into the modulation piece now and have a lot of studies that are coming online in that regard, but I will show a little snapshot of this along the way. Okay, so to paint a picture of this complexity, here is our multispectral panel that I introduced to you with the markers that I mentioned with 3 different TLS states. Here's a tonsil from a normal individual like you or I that does not have cancer. And they have a germinal center. They're encapsulated. It's very, very clear that this is a structure. And then here's what happens in multiple different solid tumors. And so we're very interested in looking at TLS across different solid tumors. We started working most heavily in head and neck and lung cancer, and now also in ovarian cancer, but we've got several projects in brain metastases and metastatic melanoma, and also now in pancreatic and colorectal cancer. But what you can see is depending on the tumor type, you get a different type of TLS that predominates. So for example, colorectal cancer patients mostly have TLS with germinal centers, and most of the patients form them. Whereas if you look at ovarian cancer patients, they have more of a range of structures, with lymphoid aggregates being more predominant depending on the site of disease, which I'll show you. Pancreatic cancer patients are similar. And we have a new study that we are just finishing up where we looked at brain mets in lung cancer patients or from metastatic melanoma. And these are often very rudimentary, but they have different activity metrics. And so you get differences even in the same site of disease, depending on the tumor that is coming into that from a metastatic standpoint. So you can see here that there's a lot of different drivers underneath each, and the percentage of patients varies depending on the type of tumor that's present and where that tumor is. Okay. So with that being said, I want to talk a little bit about our studies in ovarian cancer that were published last year. This was captained by a postdoc in the lab, Ian McFawn, who really wanted to come in and learn spatial imaging and analysis, but also really wanted to think about, spatial transcriptomics and how to apply that for in situ TLS evaluation. And so he used the panel that I mentioned to you to first just machine learn for TLS. So every green island you see here is actually a lymphoid aggregate. So we always categorize them as a lymphoid aggregate to start until we look at their sophistication. And so the point of this study was to really look at fallopian tube tumors, omental tumors, and ovarian tumors, because a lot of what's been done in ovarian cancer has been mostly done in ovary or omental tissue, with the fallopian tube not being considered. And we were really interested in how TLS might vary in these different sites of disease. And so what you can see here is that just by eye, there are more structures in the fallopian tube, because every green island is a structure, than in omental tissue. And in ovarian cancer patients, as you can see, sometimes you have only one structure, sometimes none, and sometimes there's more than that, but there's very few compared to the other 2 sites. And then these insets show you that the type of the structure is also different, which I'm going to summarize here and show you some data. But basically the composition is very different, but also the type of structure. A couple key points, and I'm summarizing a lot of data for the sake of time. You can pull this paper and read it more thoroughly. We did look in benign ovary and fallopian tube samples for people that have benign ovarian disease, no structures. And in fact, I would say even when patients are developing a stick or a serous tubal in situ carcinoma, which is a precursor in the fallopian tube for ovarian cancer, we don't really see structures come online until the stick sample has an invasive disease. We also looked at this across patient samples, but also within the same patient that had disease in all 3 sites, and the trend holds. So just if you have a patient that has a lot of TLS in the fallopian tube, that doesn't mean they have more in the ovary, right? The metrics that I'm going to show you stay the same even in matched patient samples. And one thing that we really noted, particularly comparing ovarian cancer TLS to the other 2 sites, were that they were wildly different. And indeed, they had less B cells and less high endothelial venules. They had more CD8 T cells and more Tregs within the structures, and they had reduced germinal centers and less B cell activity as a result, which I'll show you on the next slide. And here are those data. So not only was Ian able to machine learn for having a structure, but he was also able to machine learn for having a germinal center, which is this purple. So 1 is lymphoid aggregate, 2 is a TLS without a germinal center, and 3 is a TLS with a germinal center. And you can see these 3 states and the rubric that we've made to look at this in ovarian cancer. And what you see in these pie charts is that this pink part of the pie is much smaller in ovarian tumors than it is in fallopian and omental. And so all of the TLS within the ovarian are mostly lymphoid aggregates, and very few are TLS with germinal centers, where fallopian, tubonomental tissues have more TLS with germinal centers and a fewer lymphoid aggregates. And when we look at the readout functionally for this, what we see is that if we don't even consider the germinal center, B cells within ovarian cancer TLS are less active than in the fallopian tube, and then in the omental goes next. And so this is by those metrics of AID and Ki67, so proliferating cell that's also potentially going through that somatic hypermutation process. But as expected, when you add the germinal center piece in, it goes up even more based off site. And so you don't really get this in ovary. Like, we don't have very many germinal centers, so it's not significant because there's not that many. But you can see in the fallopian tube and the omental tissues, when you have a germinal center, you have then increased activity. And so this is all well and good, and this is all from spatial imaging, but we were really interested in then looking at TLS in situ in more depth, and this is when we turn to spatial transcriptomics. So we use the nanostring geomics or DSP, and we like this because you can stain exactly like you do for your multispectral imaging, but then you can get that same image with the DSP, and you can select TLS regions, but you can also segment the cell types. So if I want to know just about B cells within this TLS, I can ask about that and sequence them separately. So that was an advantage for us for this technology. And so we did the 18,000 gene WTA readout for this, and I'll show you some of these data now. And so the first thing we did is we just looked at TLS in ovarian sites, ovarian tumors, because even though there's less of them, we still wanted to know if having them were prognostically beneficial, and we wanted to generate an in situ signature of this. And so that's what we did. And this is the signature that we created by looking at TLS versus non-TLS regions. So regions that had immune infiltrate, but no TLS. And the genes here are not necessarily ones we would think of right away. Some of them are, like lymphotoxin beta. But they are definitely genes where when you look them up, they could very much be involved with TLS formation. And when we ran the survival curves using the TCGA, we saw that it was prognostically beneficial in both overall survival, but also in individuals that were under the age of 55. And this was interesting to us because basically women start to go through menopause at this time, and that's around the time when they start to then get diagnosed with ovarian cancer. So it's interesting to us that the survival benefit is in patients that are younger. And this also not only suggests maybe a hormone-based metric that's present here, which we could dig into, but also perhaps just aging of the immune system. And this survival benefit holds in multivariate analysis. So then what we went on to do is we went to go and look at the differentiation trajectory of the TLS. And so we can create this differentiation trajectory looking at no TLS to having mature TLS. And that overlaps with what we saw with our spatial imaging, that ovarian cancer patients have much fewer mature TLS, whereas omental have more mature. And that's where we started with, was the ovarian and the omental comparison. And what we also uncovered was this intermediate state of the TLS. So now we're looking at these genes that govern from immature to mature with this intermediate state in between, because it might give us indication of some of what's happening on the way for TLS development. Okay, so that's thinking more about the TLS itself and more the immune side of the TLS. We've also, for this experiment, we looked at the stromal compartment, and we were able to do that by negative selection because we had markers for all the other cell types associated with the TLS, and then the stromal compartment comes out. And we vetted that, and we saw that there were a lot of stromal differences as well based off the site of disease, right? But also just thinking about where these stromal precursors come from. And that follicular dendritic cell that I mentioned to you earlier that really governs a germinal center and governs, activity of that germinal center. It's still a question as where this comes from. And follicular dendritic cells, despite their name, they come from precursors in the stroma. They come from mesenchymal cells. And so this is where we partnered with Lon Kaufman, who's a really talented physician scientist. She treats ovarian cancer patients, but she focuses on these cancer-educated mesenchymal stem cells. And these cells are reprogrammed by the tumor. They express WT1 as one of their hallmarks, and they promote tumor initiation and immunosuppression. On the counter, if you resist that education and you're normal, or you come from the bone marrow, you can migrate to inflamed areas. And these are often the precursors of follicular dendritic cells. That's what's been shown in the literature and other supportive stroma that would help for germinal center formation. So we first started out by looking at a signature in our spatial transcriptomic data that Lon created for CMSCs. And we saw that as we moved from being close to the TLS to further away, that this signature increased. We then vetted this with another spatial panel where we looked at the TLS, and then we said, how close or far away are these normal MSCs versus these cancer-educated MSCs? And again, as you move further away from the TLS, you get more cancer-educated and less normal. So we really think that areas where these cancer-educated mesenchymal stem cells are present in the stromal compartments, it's resisting TLS development. And so we're following up on some of this mechanistically in a mouse model. But what we did for the paper is we then looked at B cell function and FDC polarization through the lens of CMSCs. So Lon can isolate these cells from primary patient tumors, and she can then we can take them and we can use them in vitro assays. And so what we saw was that if we take cancer-educated mesenchymal stem cells versus normal, the B cells don't adhere as well to these cancer-educated mesenchymal stem cells. So interestingly, I'm not showing you these data. They both can recruit the cells, but these ones don't hold on to them, which suggests that they can't bind and they can't offer that soil for seeding of the structure to start to form with the B cells. The other thing that it blunts is proliferation of the B cell. So we use conditioned media from these and showed that the B cell function is blunted by proliferative capacity, but we've also shown this in a contact dependent manner now, and we're following up on this. But finally, we also see that FDC polarization is also blunted. So if we take these cells and we push them to an FDC, you see less markers of FDC polarization for the CMSCs than you do for the normals. And we also can see this by eye visually. So we can see the cells differentiating and getting spindles, et cetera, when they're normal, but when they're cancer educated, it seems to be blunted. We also followed up and did a survival analysis in the TCGA with both signatures that I've mentioned, both the CMSC signature, high or low, and then the high DSP signature. And so what we showed is that clearly, if you have a low DSP signature for TLS, you don't do well. If you have a high DSP signature, but also have a high CMSC signature, so both the TLS and a CMSC signature that are high, it is as if that prognostic benefit that I showed you of the TLS signature that we generated goes away. But if we have a low CMSC signature with our TLS signature, patients do better. So this really does solidify that we think these mesenchymal stem cells that are cancer educated are really tuning what's going on with TLS development. Okay, in the last couple minutes, I just want to talk about then what happens when you're outside of a germinal center, okay? And this is another thing that we're thinking a lot about in the lab because you get a lot of B cells that aren't in a germinal center, as I mentioned to you. They're in a lymphoid aggregate, they're in a TLS without a germinal center, or they're just in contact with the tumor. And so one thing that we saw when we compared our transcriptional signatures of our TLS from our ovarian cancer patients is that while there's an overlapping signature, that is also prognostic across all three sites. The signature in the ovary, where there's the most rudimentary structures, the least amount of germinal centers, have hints of germinal center independent B cell responses or extrafollicular responses, so genes associated with that pathway. And so this started getting us thinking about the plasma cells because plasma cell output can happen with either process, germinal center dependent or independent. And when we look at the plasma cell readout from this study, in the omentum, where there's more germinal centers, you see a lot of plasma cells around that germinal center. But in the ovary, it's quite devoid. But when we look at the readouts across tissues, ovary tumors actually have more plasma cells across the tissue than the fallopian tube. And so that's interesting to us because they have less TLS, they have less TLS with germinal centers, and the fallopian tube has a lot of TLS with germinal centers. So one thing we're thinking is that in the fallopian tube, it could be that they're just going to more of a switched high affinity memory B cell, which is another thing that can happen with the germinal center and not a plasma cell. And we can follow up on that. But what we really think is happening in the ovary is that plasma cells are getting educated through a germinal center independent response. And so this is a background on those two responses. This is germinal center dependent, and this is the extrafollicular or germinal center independent. This is a study that was first started by my previous graduate student, Dr. Ayana Ruffin. And now she's at Emory doing her postdoc with Crystal Palos, but also now has been picked up by a very talented student, Ali Casey, who's now expanding upon these studies. And so the biggest thing that we saw and we see with this extrafollicular response is that the subsets are different, but they're also less functional than those counterparts from the germinal center. And here are those data from a recent paper this past February that highlights when you have more of these germinal center independent responses, B cells signal less, they have less antibody production, they're unable to differentiate as well to a plasma cell, and they have increased B cell specific inhibitory receptors. And so this gets us thinking a lot about the balance of these two processes, right? So we have germinal centers, but they're not the most frequent. We get a lot of germinal center independent responses. So clearly we want to think about how to either get those cells to benefit from a TLS or reinvigorate them in another way because they're not functioning properly. And all of this comes back to spatial. So I've already kind of alluded to this, but we did this metric with a spatial panel and showed that when you have TLS with germinal centers, you have more of these germinal center dependent B cells, memory B cells that come out. But in the germinal center independent response or extrafollicular response, they're mostly outside of a TLS or even in a more rudimentary TLS. And the spoiler alert here is that all of these processes, both germinal center dependent and independent, are modulated by therapeutics. And so to show you a snapshot of this, because we're working at this with multiple solid tumors, but we really see this in pancreatic cancer in a current trial that we're studying. And so a student in the lab, Charu Aurora, has really analyzed this trial thoroughly to set some foundation for her thesis. And what we've seen is that in patients that got chemo only, autophagy inhibition plus chemo, or autophagy inhibition plus chemo plus anti-PD-1, the TLS maturation goes up with each treatment. And that is enumerated here. So specifically, you can see this light tan. There's very few germinal centers at baseline in treatment-naive PDAC patients. But as we give these therapeutics, they get more of these germinal centers that evolve. And you see more activity within those germinal centers as well. So clearly, by giving different therapeutics, you're tuning these different processes. And that's something else that we also really need to understand and understand if they're both functional at that time. So to kind of wrap up, we really do think that TLS state not only dictates T cell activity, but B cell activity. And that's bore out here, looking at germinal center dependent to independent, the different subsets of B cells, which I didn't get into in a lot of detail given time. But really, we think that that is going to allow us to understand spatially then what's happening with these B cells functionally. So they're linked together. So I hope that I've convinced you that having B cells in TLS is like completing an orchestra. If you want that really beautiful sound and you want to have that harmonization, we really do think you have to have those structures or those cells present and functioning to really allow for optimal therapeutic responses. And so these are the take-home messages that I highlighted along the way, but I do want to come back to this therapeutic modulation because I think there's a lot of ways we're thinking about it. I think we're tuning oncolytic viruses and thinking about how to tune CAR T cells as we learn more about those in solid tumors. I think that can be done by using different inducers of TLS, different chemokines and cytokines that are key. I think also thinking about the stromal cells and bringing those close to the B cells and being functional through a bispecific is another way to think about it. But ultimately, I think thinking about both sides of the coin, not only what happens when you have a TLS with the germinal center, but when you don't, and how you can reinvigorate those two processes and make better not only immune responses, but specifically better B cell responses. So with that, I will thank everybody in my lab. I have an amazing group, both past and present. I tried to highlight people along the way. All of my collaborators, I only really talked mostly about the ovarian cancer work, but a lot of the germinal center independent work that I showed you was in head and neck cancer, and Bob Ferris is a really key partner in that. All of my funding, as well as all the patients and their families, because without the samples, we can't do this really important work to then go into the mouse models that we've developed. So with that, I'll stop and say thank you for your attention. If you're interested in what you heard, we have an online consortium that meets every month. It's free, it's virtual, and we talk about these topics. So please email me and we can get you signed up. Thank you so much. Oh my God.